Okay, the next topic in our series for eukaryotic gene expression is actually regulation at transcription. So basically, this is the shortest note or kind of like topic we're going to cover, but actually there's a lot in between, and of course there would be one activity that I would like you all to do, but okay, I'll tell you all later. So basically, okay, what is being regulated at transcription? We know that okay, there are controls at transcription level and also post-transcription, as you can see over here. So it will determine at transcription level whether the gene gets to be transcribed to produce your primary or your basically processed mRNA, or at post-transcriptional level, even if you did produce or transcribe the gene at primary level, will it get to be processed to produce mature mRNA for a complete production? Okay, let's see. Now, what I'm going to cover here is basically just some of the techniques, experimental techniques being basically carried out in labs by people just to determine transcription rate. And this is basically what they report for certain bacteria only, and it may vary from one bacteria to another. So there are 4 kinds of measurement, such as pulse labeling, nuclear run-on assay, polyethylene, chromosome, and study of nuclear RNA. So we will see each and every one of them. Now, for the first one, which is the pulse labeling study. So as the name says, labeling, there is a label that is being used. So either radioactive or kind of like a safe label. So the procedure, this is basically just the general procedure. So the scientists, the researchers usually like to use radioactive because you can actually determine the gene expression better using radioactive as compared to using safer label, because the safer label sometimes there would be a false positive, or it doesn't really bind that well to the cells. So of course they prefer radioactive, even though it's actually not really that safe to those who conduct it, but it's actually give good results. So once nucleotides have been labeled to the cells after about 5 to 10 minutes of incubation, and this is usually done at it depends on the condition, and also at the growth temperature of the bacteria, either at 37 or 30 or 40, it depends. So after 5 to 10 minutes of incubation, the cells will be harvested and all of the RNA isolated. So when I say RNA, all of the RNA, all different types of RNA will be isolated. So the label RNA will be hybridized to dot blot to contain DNA from the genes where the transcription is being measured. So basically, dot blot is a quantitation method to determine the actual expression concentration or expression kind of like measurement of the transcription. Now, what happens is that synthesis of the RNA is usually measured by the addition of the radioactive nucleotide, which is uridine labeled with tritium, and measured by the incorporation of the radioactive. So this method minimizes degradation of newly synthesized RNA, and this is done by exposing the cells briefly to the label uridine, because if it's exposed for a long period of time, then probably the newly made or newly synthesized RNA will basically probably not going through thorough degradation. So the right way of measuring transcription rate and also transcriptional control has been more or less said to be better in this study, but there is of course a limitation in terms of application, as it requires very short labeling time, 5 to 10 minutes only. This is to minimize degradation, and the short time pause is sometimes insufficient to measure rate of transcription in many other species. In E. coli, in Bacillus, I actually forgot the other bacteria. It has been proven to be okay, but not so for some other species. Now the second one is the nuclear run-on assay. So the procedure is as such, as you can see from your screen and from your notes. So the first one is that your nuclei is isolated from the cells, and of course, radioactive labeled nucleotide is added to it. So incubation period for this kind of assay is longer, after about 1 to 2 hours of incubation. Again, at the temperature whereby the bacteria grow. Afterwards, RNA will be isolated from the nuclei, okay? And this would include all the labeled RNA produced by the transcription of the run-on, okay, to the end of the gene. So the labeled RNA, okay, will also be hybridized to dot blot to contain DNA from the genes, whereby you could actually quantify the transcription rate and also the intensity plus the concentration. So more or less, this is how nuclear run-on assay is being done, as you can see. So in your cell, okay, so we know that this is the nucleus, okay, this is the cytoplasm. So what happens, okay, is that basically polymerase transcribing, okay, a gene, okay, is being attached over here by the labeling procedure. So isolate the nuclei, okay, add the label nucleotide, okay, and then basically you do a polymerase run-on, which is basically done by the incubation. So you isolate the label RNA, hybridize to dot blot, okay, and then you basically see if it is transcribed or not, and then you could quantitate it. So this kind of assay is based, of course, on fundamental that a lot of precursor ribonucleotides are present in cytoplasm, which is mostly true. And by removing the cytoplasm and isolating just the nuclei, we'll also remove much of the unlabeled ribonucleotide, so that we actually remove a lot of the unnecessary particles as well. So RNA polymerase would then stop transcription due to lack of ribonucleotide, but remains associated with DNA because there is no repressor and whatnot. But if the labeled ribonucleotide is being added, RNA polymerase will resume transcription to produce RNA transcript incorporated with labeled nucleotide. So it could later be detected by hybridization, okay? And this would correspond to the DNA, which is similar to the first method, which is the pulse labeling experiment. So this method would allow quantification of the transcription of genes, actually much better as compared to pulse labeling experiment. Now, the third one is the polythene chromosome. Ever heard of polythene? If you haven't, Google it up, okay? See what is a polythene chromosome. So this kind of method is able to directly visualize increased level of transcription. Interesting, isn't it? You could actually just view it, observe it directly. So the idea came from salivary gland of Drosophila, of course, okay, the housefly. So basically, upon several amplifications of the chromosomal DNA after PCR, so you will get polythene chromosome. What we mean of polythene, okay, is that basically, well, go and look it up, okay, but okay, after you have done several amplifications, okay, there is a puff effect. Puff, which means that it's kind of like expand. I think I have it in the next slide, where the condensed or decondensed DNA area results in chromosomal expansion. Kind of like when you eat too much, okay, you get either bloated or your tummy stick forward because it is too full. Kind of like that. So the puffs represent sites of intense transcriptional activity, which could be used to hybridize to label ribonucleotide, much similar to the first method. So these sites are suggested to contain gene encoding proteins due to production of new proteins from the Drosophila. So you can see that, okay, this is the polythene urethane from Drosophila melanogaster, general one, okay, but okay, if transcription, okay, is actively basically occurring, you can see that there is a puff. This is the puff effect, okay, which means that it expands, so that you can actually see, okay. So by right, you would see, okay, some bands there from the chromosomes, okay, but you couldn't really see it because it has expanded too much. So you know that a lot of transcription has occurred over here. So this is also, and of course, okay, it is a direct observation, direct visualization of the transcription occurring. It's just that if you were to measure the rate of transcription to really quantitate it, then you need to apply either the pulse labeling or the nuclear run-on assay. Alright, so I think I will stop here. Okay, move on to the second video.